Antibodies protect you, but antibodies kill. Autoantibodies can make you very ill. Antibodies coat pathogens, they coat the infected cell. With NK cells and phagocytes, they give those microbes hell. When antibodies fix complement, the shit really hits the fan. You'll be blown to bits, little microbe. Run away if you can. You take a set of beta strands, you get a beta pleated sheet. Fold the pleated sheet in two, then the barrel is complete. Clip it with a disulfide bond, and you have an IG fold. Found even in archaebacteria, this domain is old. Loops make connections between the beta strands. If you want to make an FAB, please use both your hands. The loops are like fingers. They stick out at the top. Complementarity determining regions, the cream of the crop. 3 fingers from the heavy, 3 fingers from the light come together to create an antigen binding site. Bring the fingers close together, you get a cleft with a purpose. Splay the fingers widely, you get a protein binding surface. Antibodies protect you, but baby, antibodies kill. Autoantibodies can make you very ill. Or you Y-shaped globular proteins with a name that Ehrlich gave. If the good doctor could hear this Doggerel, he'd be turning in his grave. Although the functional properties of antibodies were described first in the 1890s, the structure of antibodies was only elucidated about 70 years later, and we now know that antibodies are made up of 2 protein chains. One chain is called the heavy chain, and the other is called the light chain. And then the antibody can be proteolytically cleaved into fragments, and one fragment, which includes the entire light chain and part of the heavy chain, is called the FAB fragment, and this one binds antigen at this end, whereas the other part of the antibody, the tail, is called the FC portion. Now, the functions of antibodies, we said antibodies coat pathogens, they coat the infected cell. So pathogens which might not be sometimes recognized directly by phagocytes can be coated by antibodies. So here you can see you have an antibody coating a bacterium, and here's another set of antibodies coating a virus. And these coated pathogens can be internalized by the phagocyte, a neutrophil or a macrophage, because these cells have receptors for the FC portion of the antibody that coats the pathogen, and they can thus internalize the pathogen and destroy it within phagosomes. So this property of antibodies is called opsonization, wherein the antibody helps pathogens be internalized by phagocytes. The other thing the antibodies can do is that they can coat infected cells. So if you have a virally infected cell, and here we have a spike protein of a virus on the surface of the infected cell, the antibody can come and bind to the spike protein and then recruit from its FC portion. It can recruit cells called NK cells, and the NK cells have receptors which bind the tail of the antibody, activate the NK cell. The NK cell then secretes stuff that will cause the death of the infected cell by apoptosis. So this process by which an antibody coats a virally infected cell and then recruits an NK cell to kill the infected cell is called ADCC, or antibody dependent cellular cytotoxicity. The other function of antibodies is the fixation of complement. So when an antibody is bound to a microbe, the first component of complement can be recruited to bind to the antibody, and this sets in motion a cascade of events, proteases numbered from 1 to 9. So we have 9 different complement proteins, which then this ends with the process of the formation of a pore, with the C9 protein forming a pore. And then salt and water can flow into the microbe through the pore, and then the microbe basically gets blown up. So this is a major function of antibodies, the fixation of complement. Turning to the structure of antibodies, so the basic unit of the antibody, each domain, as we'll show in a second, is made up of a beta-pleated sheet. So this is a beta-pleated sheet. And a beta sheet is made up of beta strands. And the beta strands are joined by loops, as you can see here. And these beta strands together form the sheet, which when folded over can form this structure here called a beta barrel. Okay? And if this beta barrel is held together by a disulfide bridge, it's called an Ig fold or an Ig domain. And this is the basic unit of the antibody. It's found in many other proteins, but it was first discovered in antibodies. This basic modular unit, the Ig fold or the Ig domain. So if you were to look at an antibody, it's made up of multiple Ig domains. So this domain here, for instance, in the light chain is called the variable domain of the light chain, and it's called variable because it varies from one antibody to the next. And here this would be the variable domain of the heavy chain. This is the constant domain of the light chain. And then the heavy chain, we're showing an IgG molecule here, has 3 constant domains. We have CH1, CH2, and CH3. And you'll note that each of these domains is a beta barrel, basically made up of a beta sheet that's folded up. Now, when we take this further and try to understand how antigen is recognized, what you would notice is that we do have these 3 fingers that stick out at the top. So that's what we described the antibody as having. So these fingers here, the 3 you see from the heavy chain, all these fingers are called CDRs for complementarity determining region. See, we have CDR1, 2, and 3 from the heavy chain, and then we have CDR1, 2, and 3 from the light chain. So you have these 6 fingers sticking out. We have an identical structure on the other side. Just drawn this over here. And what you could see here is that maybe an antigen, and you see a little cleft that's formed here. There may be an antigen that is shaped like this, that comes and binds tightly and is held within this cleft. So the very same antigen will be bound on the other side by the other FAB as well. Sometimes antibodies may have these CDRs which actually stick out and splay out and form a surface. So here we have these 6 fingers splaying out to form a surface. And here you can visualize maybe there would be a larger antigen that can bind to this antibody. And this would be perhaps a large protein. So this would be an antigen. But here the antigen is being recognized by the splayed out fingers formed by the CDRs of the antibody. A very similar, I would say an identical event would happen on the other side because it's the same 6 fingers on the other side as well. So we'd have the same antigen also being bound by the other half of the antibody. And this, in essence, is a summary of how antibodies recognize antigens. Paul Ehrlich was unquestionably the first person to think about the existence of an immune system. He was the first person to think about how immune recognition occurred. He was the first to consider diversity in the immune system. And he also was the first person to think about autoimmunity and tolerance, about the surveillance of tumors by the immune system, and he created the world's first chemotherapeutic. So while I'm not a great believer in heroes, if there's someone one must really think of admiring in the past, for me, one of those people would be Paul Ehrlich.